This video provides an intro to the basics of protein structures: primary, secondary, tertiary, and quaternary. You'll remember from previous work that when you have two amino acids, and this shows a beta-ene charged form of the amino acids, two hydrogens from the nitrogen and the O from the carboxylic acid get together and form water in a condensation reaction that also makes a peptide bond. So this bond here hooks into the carbon and here's the double bond O. The primary structure of a protein or a small polypeptide is shown here. The convention is always to write it from the N terminus to the C terminus, and you can see the series of amide bonds. That primary structure is just the beginning of protein structure. Secondary structure is the localized conformation of the polypeptide backbone, and we'll look at two types in this video: alpha helices and beta sheets. And then tertiary structure is how those various components of secondary structure fold together and create a three-dimensional structure for the entire polypeptide. And finally, if several polypeptide chains get together to form a larger protein with multiple of those subunits, that is known as quaternary structure. Proteins vary widely in size and composition. You can see the very small protein, insulin, which we've looked at before with only 51 amino acid residues, but it has two polypeptide chains. A much bigger protein, myoglobin, has only one chain, so it's hard to predict the number of chains from the number of amino acid residues. It is true that the really huge ones with 5,000, 6,000 amino acid residues tend to have more chains and more subunits that are put together in quaternary structure than the smaller ones. Now we're going to dig into the structure of the amide bond, and it has two resonance forms. The nitrogen lone pair can move into the carbon to make this double bond and a positive charge on the nitrogen, and one of the pairs of electrons in the double bond moves out to form a lone pair out on the oxygen with a minus formal charge. The consequence of this is that the geometry changes. Here we have a trigonal pyramidal nitrogen and a trigonal planar carbon. Here we have a trigonal planar nitrogen and a trigonal planar carbon. Reality looks much like the second picture. For a trigonal planar compound, you'd expect 120-degree angles, and here you see that the carbon does have roughly 120-degree angles, but so does the nitrogen. So it looks like this all is one big flat plane with the nitrogen, carbon, and oxygen all in that plane. And rotation can occur around this bond and around this bond, but there's restricted rotation around the peptide bond because it has some double bond character in the resonance form. This little rectangle is known as the amide plane, and you're going to see that as a schematic in later diagrams. Here you go. There's an amide plane, and there can be rotation around this carbon alpha, and then there's another amide plane, and then rotation around this. Note that the side chains aren't shown here, the hydrogen or the side chain that's hanging off that alpha carbon, just to simplify the idea that a peptide or a polypeptide is a series of these planar elements, sort of like a bracelet with flat rectangles making up the individual joints. Here's an even longer view, which does show some side chains, and very commonly the oxygens try to stay away from each other. Here's a picture of two important angles in a protein. The psi angle is here, and the phi angle is here. So relative to the nitrogen to the alpha carbon, that's the phi, and the carboxylate carbon to the alpha carbon, that's the psi angle. and they tend not to like to rub those carbonyl oxygens together. They both are rather large, so there's restricted rotation. It's not freely rotating all of the way around the chain of the bracelet. Another really cool thing that happens is that the hydrogen bond acceptors on the carbonyl oxygen and the hydrogen bond donor out here on the nitrogen like to get together, and they can form all different types of helices where those two, where a hydrogen bond can form between members of subsequent residues. So the most common one that we're going to focus on is the alpha helix. In the alpha helix, the oxygen gets together with a hydrogen, 1, 2, so 1, 2, 3, 4 nitrogens away. So it's known as the 1,3 helix. It's the fourth residue up that forms a hydrogen bond. Three-dimensionally, the alpha helix is the one shown here in the middle, and you can orient yourself a little bit and find the carbonyl oxygen, and you see that it's hydrogen bonding to the nitrogen, the hydrogen that's on the nitrogen, and if you go around the chain, this is 1 nitrogen, 2 nitrogens, 3 nitrogens. It's the fourth nitrogen up from this carboxylate C double bond O. Schematically, alpha helices are shown as ribbons. We'll see that we don't even see the atoms anymore in future slides. You'll just see a ribbon, and that indicates that there's an alpha helix there. Here's what they look like in a space-filling model and a ball and stick model. There really isn't a lot of space in the alpha helix. The inside is completely taken up by the backbone, and the side chains protrude out. And I wonder if you can pause the video and see if you can identify some of those side chains. This is sort of an abrupt transition, but you can see the way 3 different helices look in a very schematic form where you see just the ribbon. This is the alpha helix, and it actually moves. You can turn it around in space. You can look down the center. Of course, when you saw the space-filling model, you realized that there's no space there, actually. It's all taken up by the backbone atoms. And the hydrogen bonds form along these 1 to 4 connections up the chain. Beta sheets are the second type of protein secondary structure, or a local folding of the amino acids based on hydrogen bonding interactions. And beta sheets are schematically represented by big arrows, and then the part here is irregular secondary structure, where they're not hydrogen bonding networks in regular arrays the way they are in alpha helices or beta sheets. This is an anti-parallel sheet, and here is a parallel sheet, and we'll look more closely at the structure of both of those. Here's an anti-parallel sheet, and you can see that we're starting at the N terminus here. Now you might be confused by the fact that the O is sticking out in what's supposedly the N terminus, but if you look closely, this is the amide linkage. So here's the alpha carbon, and this would be the N, the nitrogen end of this particular piece of the chain. So this is the N terminus, and down here, after the amide bond, there would be an alpha carbon and then a COO minus if it was truly the end of a chain, or at least the end of this little piece of the chain. So N to C running this way, and opposite that, N to C running this way. That makes it anti-parallel. And you can see a really awesome lineup of hydrogen bonding interactions here. The H that's hooked to the nitrogen is right across from the carbonyl oxygen and its lone pairs. So there's a perfect hydrogen bonding opportunity, and every single one of them lines up that way. It's a really, really cool structure. In the parallel sheets, you can see that if you start N here and run to C, and N terminus and run to C terminus this way, the hydrogen bonds don't line up as precisely opposite each other, but they're still excellent hydrogen bonding interactions at these angles here. This is just a close-up of the anti-parallel structure, And I want you to think about where the side chains are. It turns out that they stick sometimes up from this sheet and sometimes down from the sheet. And you'll see that in later slides. And here's the parallel, again, a close-up view. This is another view of along not just 2 strands, but several strands, 3 strands of anti-parallel and parallel. And this is what makes it a sheet, because you can keep adding more strands and they keep fitting into this structure. So what does that beta sheet look like in a better 3-dimensional representation? Here you can see one. It's really, really neat. And if I hold it this way, you might be able to tell what the amino acid residues are. Do you see that the ones sticking down have only a hydrogen and another hydrogen? So those are all glycine residues. The glycine side chains are all sticking down, and sticking up you see a bunch of CH3s here, and I hope you can recognize CH2OH and figure out what amino acid that might be. So it's a neat structure, and you can tell now that in the secondary structures, both beta sheets and alpha helices, the backbone gets buried in the internal part of that structure. And what's important, what sticks out into the environment, whatever the environment is the side chains. That's why we focus so much on whether those side chains are polar or nonpolar, because that will control where this sheet likes to be. If I change it to a rainbow view, it's much easier to see that it runs one direction and then the opposite direction, up and down in this beta sheet. And if I make it even more schematic, this is the cartoon representation. You can see sheet running one way, sheet running the other way. And finally, we can look at the actual hydrogen bonding, and that makes it really clear that this is an anti-parallel sheet because of the neat little hydrogen bonds. It's not quite as sexy as that moving picture, but here's a cool picture of carboxypeptidase A and a beta sheet from that enzyme. You can see the backbone structure and the side chains hanging out here, and I love the space filling view where you can see how bulky those chains really are, and in this case very hydrophobic. Now we're just going to look at some representations of whole proteins. This is triose phosphate isomerase, and first you see the space filling model, which is neat looking but doesn't necessarily let you dig into it very easily, especially if you can't twist and turn it around in this image. This is a really interesting way to represent a protein. What's shown is the polypeptide backbone, and each vertex is an alpha carbon. So this is a line from one alpha carbon to the next alpha carbon is what you're following in this representation. You can sort of see some alpha helices in that, but I think the easiest way to see alpha helices is with the ribbon diagram or the cartoon diagram, where it's really obvious that this is a big region of alpha helix. And I think there are a couple of beta sheets on the inside of this protein. Here are 4 more proteins, and I'd like you to pause the video and try to decide whether each one is mostly alpha helix, mostly beta sheet, a mixture of the two, or mostly irregular secondary structure. Okay, I hope you were able to figure out that this is almost all alpha helices, and this is actually growth hormone. B is gamma beta crystalline, and it's full of beta sheets and doesn't have a single alpha helix. The flavodoxin here has a mixture, this is very common, a mixture of beta sheets and alpha helices, and a fair amount of irregular secondary structure as well. And tachystatin, this little one here, has a couple of beta sheets, but is mostly open, irregular secondary structure. This is a really cool image. It's a proposed time sequence of how a protein might end up folded. So the primary structure is synthesized and it's just a blob, and then pieces of that blob start to self-assemble into the hydrogen bonded elements of alpha helices and beta sheets, and then those pieces start to fold further based on intermolecular interactions. That's why we're beating so hard on intermolecular forces in this course, and assemble more, and finally end up in a tightly coiled form. And this form is the form this protein will always take. So whatever this primary structure is, it ends up being folded like this every time, and that's a miracle. I think it's amazing and really beautiful that we always end up with the same tertiary structure for a protein, just based on it going into water with a certain amino acid sequence, and all those amino acids either wanting to be near water or not wanting to be near water, and wanting to hydrogen bond with each other or not wanting to hydrogen bond with each other. The last thing I want to look at is where the amino acid side chains end up, or where they point, depending on what a protein likes to do. So this is myoglobin, a globular protein that floats around freely in the aqueous cytosol. And you can see in this image that the green colored species are the nonpolar or hydrophobic amino acids. And if you look closely, they are inside the protein. They're mostly buried inside the protein. So the way it's folded, it puts those in the innards of this glob. And the red here is the polar or charged side chains of amino acids. And you can see that those point out toward the water because they're hydrophilic and they like to interact with water. And these nonpolar ones tend to like to interact with each other instead. So this phenomenon is extremely common, and you'll see that membrane-spanning proteins tend to put the nonpolar amino acids in the part of themselves that goes inside the membrane, and they put the polar or hydrophilic pieces in the part that sticks out into the cytosol. And we'll see that over and over, that the hydrophobicity of the side chains on the amino acids of a polypeptide or a protein control the way that molecule behaves.